Hello, my name is Taylor Quirley, and this is my presentation on the disorders of carnitine transport. So carnitine transport is an important part of the metabolism, and this is just a big picture showing of it all. So carnitine primarily functions to um, bind to fatty acids, which allows it to go across the inner mitochondrial membrane, where that fatty acid is then delivered to go into beta oxidation. And once it goes into beta oxidation, it goes through the TCA cycle where it produces products like NADH, FADH2, which can then go to the ETC to produce ATP. And so beta oxidation, uh, this occurs primarily in times of starvation, and carnitine is so important just for beta oxidation to even happen, where if you remove carnitine, beta oxidation can't happen, and then there's a greater dependency on glycolysis. So this is just an overview of the important enzymes, important transporters that's within this carnitine system. So first you have your free carnitine that's outside of your cell, and this comes from diet or from other processes that might make it. And so this binds to your carnitine transporter, although otherwise known as the OCTN2. So this binds carnitine with a high affinity, and it allows it to enter into the inner plasma. And so carnitine, once it hits your outer mitochondrial membrane, it'll encounter this enzyme, a carnitine palmitoyl transferase 1, and this enzyme is responsible for binding a fatty acid chain to carnitine so that it can enter into the inner mitochondrial membrane. And this cannot happen, like this fatty acid chain cannot enter unless it has carnitine attached to it. And once it enters here, it can then enter the inner mitochondrial membrane via this carnitine acyl or carnitine translocase, which exchanges acylcarnitine to enter the inner mitochondrial membrane for carnitine, which this free carnitine will exit. So this free carnitine is formed by the reverse reaction of CPT1, which is carnitine palmitoyl transferase 2, which takes your formed acylcarnitine that can enter into this inner mitochondrial membrane, and it'll separate it into carnitine to be exported out and acyl-CoA to be sent into beta oxidation. So this is the full system, the full working mechanism for the entire process. So you have carnitine that's outside the cell, that's provided for through the diet. It's transported through your this OCTN2 transporter. Here's your carnitine within the cell. It encounters CPT1, and this enzyme will bind carnitine to your acyl fatty acid, and it's then able to enter through your CACT this translocase, which exchanges it again for carnitine, and then CPT2 breaks this down into acyl-CoA or acetyl-CoA and carnitine to be sent out for beta oxidation. And so again, once this hits beta oxidation, that acetyl-CoA is turned into NADH, GTP, FADH2, which are sent to the electron transport chain, and ATP is produced from this. So before we get into the deficiencies with the carnitine shuttle, I wanted to just try to define a few of the resulting disorders from the various deficiencies. So first we have this form of hypoglycemia, and this is just reduced glucose levels, and this is primarily due to an increased need for the energy dependence on glycolysis, since the deficiencies we're going to discuss will interfere with beta oxidation. And so hepatic encephalopathy, this is just inability of the liver to filter certain toxic substances that might be built up due to the deficiencies. And we also have skeletal and cardiac myopathy, which here's just an image of it. These later deficiencies can cause just weakness in muscle tissues like the heart or other skeletal muscles. Then we have this condition where it results in higher concentrations of ammonia in the blood, and then this is kind of a byproduct of the cardiac myopathy, where just weakness of heart can cause irregularities in heartbeat and rhythm. So the first deficiency we're going to talk about is called primary carnitine deficiency, or it's also known as OCTN2 deficiency. So this is where it's kind of the beginning of the whole process, and it's where this transporter right here either it's not there or its functionality is not there. So carnitine isn't able to enter into the membrane at all, and so carnitine there is not able to bind with any fatty acids, so no fatty acid can enter for beta oxidation. And since this carnitine is here just outside of the tissue, there's really nowhere else for it to go but to be filtered out through the urine. So high levels of carnitine in urine might be an indicator of this deficiency. And so some resulting disorders from this include hypoglycemia, and that's just due to greater energy dependence on glycolysis, which needs glucose, and then weakness of muscles through myopathy. So this deficiency kind of takes place in step 2 of this process. So your carnitine is able to enter a perfectly good transporter, so now it's within the cell. So once it travels to the outer mitochondrial membrane, where CPT1 usually is functioning correctly, instead in this deficiency it's not. So since CPT1 is the enzyme that catalyzes the reaction of combining carnitine to a fatty acid, this reaction doesn't happen. And so because carnitine is not attached to this fatty acid, it can't enter into the cell at all. So again, beta oxidation isn't able to happen, and you again have this hypoglycemia as you're depending on glycolysis more for energy. But in this case, since carnitine is able to actually enter the cell, you'll actually have elevated levels of plasma carnitine, which makes sense. And another interesting thing is CPT1 actually exists in 3 different isoforms in humans. There's one in the liver, one in the heart, one in skeletal muscle, but the only one that there's actually an observed deficiency in is the liver isoform. So this is the next step just in this process. So carnitine enters through this transporter, then it's able to interact with CPT1. So there's actually a successful formation of your acylcarnitine. But this deficiency is this translocase, which allows this acylcarnitine to enter into the inner mitochondrial membrane and the free carnitine to exit. So with this, again, you're similar to CPT1, like your fatty acid chain is unable to enter, which prevents beta oxidation from happening as there's no available fatty acid to oxidize, and you're relying again on glycolysis. So you see your hypoglycemia. It actually produces hyperammonemia, and then you have weakness in the heart due to this. And one interesting fact about this is this deficiency is actually most common in the neonatal period of infants. So that's within the first 28 days of an infant's lifespan. That's when this is most oftenly seen. And because carnitine is able to be formed, or your acylcarnitine is able to be formed from carnitine, as opposed to the CPT1 deficiency, you do have a low plasma carnitine levels within the cell. This is the last step of this reaction, this last deficiency. So to walk it through it one more time, your carnitine enters through your transporter, interacts with CPT1 where it forms your and it binds with a fatty acid, so it forms your acyl carnitine. Then here we have a working translocase, so the acyl carnitine is able to enter while this free carnitine can just exit. And so CPT2 is, just as a reminder, it catalyzes the reaction that separates your carnitine from your acyl group, which is supposed to be sent into beta oxidation. But in this deficiency, your acyl carnitine just kind of sits inside your membrane without being able to be separated. So this hinders beta oxidation, and which prevents the energy needed to get from it. So again, you're dependent on glycolysis, and it causes weakness within the heart, and because the heart relies on fatty acids for energy and irregular rhythms. And this will actually cause a buildup of acyl carnitine in your blood and tissues, and this is often seen in adults with this condition, and it can be triggered by prolonged exercise, which I thought was interesting. So treatment for all these deficiencies are actually pretty similar for all of them. For your transporter deficiency, one of the best things for it is just your dietary carnitine supplementation, just to increase the amount of carnitine into your diet, just so that hopefully excess carnitine can make its way into the cell somehow. And for all of these, it's recommended to really avoid fasting, and that's because in a fasting or starvation setting, you're relying on beta oxidation to produce energy, and if you can't do beta oxidation, then you can't get the right energy you need. So avoiding fasting was a recommended one. And then another kind of dietary addition is low-fat diets with medium chain fatty acids, and that's just to decrease the need to break down heavier, fatter fatty acids. That's kind of harder to do when you're struggling to send things through beta oxidation. So these are my questions, just to take home and maybe research a little bit. So my first question is, why are low-fat and medium-chain triglycerides a dietary treatment for some of the disorders presented? And then CPT1 and CPT2 form and break acyl carnitine so it can be transported into and out of the mitochondrial membrane. Why is this necessary, and what would be the effects if fatty acid could pass through freely? Here are my references. Thank you.